All right, in chapter 17, part 1, we're going to talk about the idea of going from the gene to protein. So we're going to start talking about transcription, taking the information in the DNA and getting that to the messenger RNA. So the main idea here is that genetic information in the DNA can be accurately copied and can be translated to make the proteins needed by the cell. All right, so now just in general, the bridge between the DNA and the protein is the construction of the messenger RNA. So the DNA contains, of course, the genes that make us who we are, give us our eye color, hair color, skin color, those types of things. And the characteristics that we have are the result of the proteins that come from those genes during the process of transcription and translation. Okay, so the main question here is somehow the information content in the DNA strands, the specific sequence of the nucleotides of the DNA, needs to be turned into protein. So how does this information determine the organism's appearance, and how is the information in the DNA sequence translated into that specific trait? So that's what we'll deal with in this part, and then the future parts that we're going to go through. Okay. So RNA, of course, is a single-stranded compound that carries the message from the DNA to the ribosome so that the protein can get made. And recall that DNA contains the A, T, C, and G, like we see right here, and then the RNA contains A, U, C, and G, so that uracil gets substituted for the thymine. Okay, and the order of the base pairs is what's going to determine which amino acid gets laid down, and of course which protein that we get from the 20 different amino acids that we have available to choose from in our bodies. Okay. So RNA is used because it's a way to protect the DNA from possible damage. So if we were just using the regular copy, that could be a problem. So if you think about like making a backup copy of a paper or something that you're writing on, and then you work from perhaps something on a jump drive or something in the cloud or something like that, you've always got like that master copy that you can go back to. And RNA is really a way that you can, preserve the information content in the DNA, in the nucleus, preventing damage. And it's also a way that we can make multiple proteins simultaneously. So if we look here at this polyribosome, this is just an example of what we mean by being able to make multiple ones simultaneously.
As soon as a messenger RNA molecule is transported from the nucleus to the cytoplasm, ribosomes begin to translate the sequence into amino acids. Typically, many ribosomes translate the mRNA simultaneously. Each ribosome begins at the 5' end of the mRNA
and so you can see that this is one messenger RNA molecule, this blue thing, and these green things are the ribosomes. So multiple ribosomes are going to hop on, and then these green things that we're seeing right here, these are the little kind of strands that are coming off of the blue strand. Those are the proteins that we're making.
Steadily towards the 3' end. New ribosomes attach to the 5' end at the same rate as the previous ones move out of the way. These multiple initiations allow the cell to make much more protein from a single message than if one ribosome had to complete the task before another could begin. When a ribosome reaches a stop codon, the ribosome and the new protein dissociate from each other and from the mRNA. This electron micrograph depicts a membrane-bound polyribosome from a eukaryotic cell.
Okay, so that's that video. All right, so now as far as the messenger RNA is concerned, that's the vehicle that's carrying the information from the nucleus, from the DNA to the ribosome in order to make the protein. And you have RNA polymerase, of course, the thing that we've been talking about a little bit in class. This is what's going to pry apart that DNA, and it's going to join the nucleotides together, of course, in this 5' to 3' end, adding to that free 3' end, just like we do when we're manufacturing DNA, only this is the RNA. And that RNA polymerase doesn't need a primer like the DNA polymerase does. So it just, once it hops on, it can get going. The DNA polymerase, of course, is going to need an RNA primer to get going, and all of the kind of the details that we've talked about with that so far. Okay, so transcription and translation, this is the process of going from the gene to the messenger RNA. That's the transcription part. And then the translation is going from the messenger RNA to the protein. So if you recall the central dogma of biology, we have the DNA to the RNA to the protein. Okay, that's the central dogma, and then this really is to the phenotype. Okay, whoops. And that phenotype, of course, is our outward appearance and what we look like. Okay, so that's that central dogma of biology that we've been talking about, and transcription and translation are a part of that. So the messenger RNA produced during the transcription, this gets read at the ribosome, results in the manufacturing of a protein or a polypeptide. That polypeptide, of course, consists of the amino acids, and the sequence of the amino acids is determined by the sequence on the DNA, and that's going to determine, of course, the primary, secondary, tertiary, and then perhaps the quaternary structure of the actual protein molecule. Okay, so with transcription, what we have is the gene. This is going to determine really which, or the sequence of the bases is going to determine what the messenger RNA molecule looks like. And on the DNA, one of the DNA serves as the template. That's the part that's going to get copied. The DNA, of course, is read from 3 to 5, okay, so that we can synthesize it from 5 to 3, like we've been talking about. And it's important to note that not all of the DNA codes for proteins, okay? So there are some non-coding regions, and we'll get into those a little bit more here in a minute. But as far as the segments of the DNA that bind some of these transcription factors, okay, these are the things that are necessary in order to express certain genes in certain cells, because all cells have different combinations of these transcription factors that turns on different genes. And you can see we have the start here, and we've talked a little bit about this in class, the TATA, the TATA box. That's the part where the first part of the RNA polymerase is going to bind, and then we have a bunch of these TF, these are transcription factors. We have a bunch of things that are going to join on, and then you get this basically this transcription initiation complex, this entire kind of mass of different proteins and the RNA polymerase molecule and transcription factors and all of these things that are going to bind to this promoter sequence that are going to be responsible, and there's going to be some enhancers that are going to get the process going. It's going to bend this whole thing around and get the process really manufacturing the messenger RNA kind of rapidly until we get to this termination sequence, which is also a part of the DNA that's really non-coding. And that's what these things, these are some examples of non-coding DNA, okay? And these things are involved in the things that we were just talking about here. So other functions of the non-coding DNA, they're involved in regulating the gene expression. Certain little messenger RNAs, or excuse me, certain RNA molecules can bind to it and prevent the transcription of the gene and silence certain regions of the gene and make sure certain genes don't get turned on, etc. And others code for transfer RNA molecules. These are important in bringing the amino acids to the ribosome during the process of protein manufacturing of the protein and so forth. And then there's others, just kind of as a little aside here, the telomeres. Okay, so this would be an example. Back up here just a sec, change the color a little bit here. This would be an example of a telomere. This is the end of the DNA, okay? So you can see that there's a little telomerase molecule right here, this green thing. And this is going to bind to a region of the DNA here. So that what it does is telomerase, of course, is an enzyme. It's going to extend the end of the DNA. So that's what you see here in the pink. And what it does is that, of course, we know that we go from 5' to 3' with the DNA polymerase. So it allows us to make the DNA at the very end. So if we didn't have this telomerase, if we didn't have this region here for the DNA polymerase to bind to, we would continually shorten the DNA and we would start to lose information. So this telomerase binds to the end of the DNA, a non-coding region, and it just allows us to kind of extend the length of these things, okay? So if you have some more questions about this in class, of course, we can go over it. Just make sure you write it down and I'll talk about it with you. All right, so the transfer RNA now, this is what's going to bring the amino acids to the actual ribosome. And like the messenger RNA, these things get made in the nucleus and they're used over and over and over again. And what they do is these things are going to bind to the particular amino acids. And then the other end, you have the binding end, and then you have this anticodon end, okay? In this anticodon part right here, right here, this is the part that's going to bind to or match up with the messenger RNA to ensure that the correct amino acid gets added in. Okay, so this anticodon, of course, is going to be complementary to the messenger RNA molecule. It's going to bind the messenger RNA according to the code and ensure that the correct amino acid gets added in. So a particular question might look something like, where does the transfer RNA activating enzyme attach the appropriate amino acid to the transfer RNA molecule? Okay, and what you would see from this is you have the choices A, B, C, or D, and I'm going to let you guys go ahead and answer that, and then we'll go over what the answer is in class, okay? So keep that in mind. You can back up and kind of look at the picture right here and see where you think that adds, okay? So with transcription and translation now, in eukaryotes, one of the things that's important to note is that once the genes get transcribed, they're not ready to use because there is some intronic DNA, some DNA that doesn't code, that's not needed for the making of the proteins. And the initial transcript, as it's called, is often much, much larger than the final messenger RNA that's actually going to carry the message. So you might have something that is on average about 8,000 base pairs long, and once it gets modified, it might only be 1,200 base pairs long. Okay, so you have these large non-coding regions, these things known as introns that you can see right here. These things, these pale kind of peach-colored things, these things are going to get spliced out, and then these exons, this is expressed region. Okay, that's the exon. They're going to get spliced together. And these introns here, these intervening regions, as they're called, intron for short, these things are going to get chopped out. So we're going to get rid of that. And that's what's going to allow us to kind of express this gene that we see right here. And then we're going to modify the ends. We have a 5' cap and a poly A tail, and that poly A tail prevents it from being degraded in the cell, and the 5' cap is going to facilitate attachment to the ribosome. So you're going to want to make sure that you know those things and what those are used for. Okay, so some of the untranslated regions of the exons are saved because they have important functions, for instance, like the ribosome binding, like we were just talking about with that 5' cap. So, there are some regions that really aren't going to code for the protein as much as they are going to facilitate attachment to the ribosome. All right, so here's a video now that I want you guys to watch. All right, so let me get this thing called up here. This is the RNA splicing, of course, like it says. This is what I was just talking about.
As DNA is transcribed into RNA, it needs to be edited to remove non-coding regions or introns shown in green. This editing process is called splicing, which involves removing the introns, leaving only the yellow protein coding regions called exons. RNA splicing begins with assembly of helper proteins at the intron-exon borders. These splicing factors act as beacons to guide small nuclear viboproteins to form a splicing machine called the spliceosome. The animation is showing this happening in real time. The spliceosome then brings the exons on either side of the intron very close together, ready to be cut. One end of the intron is cut and folded back on itself to join and form a loop. The spliceosome then cuts the RNA to release the loop and join the two exons together. The edited RNA and intron are released and the spliceosome disassembles. This process is repeated for every intron in the RNA. Numerous spliceosomes, shown here in purple, assemble along the RNA. Each spliceosome removes one intron, releasing the loop before disassembling. In this example, 3 introns are removed from the RNA to leave the complete instructions for a protein.
Okay, so that's going on in your cell, and that's RNA processing, and that's how you get rid of the information that doesn't code for protein, and you splice together the ones that are coded for protein. And this process is unique to eukaryotes. It does not occur in bacterial cells. Okay, all right, and then what we have is one more video that I want you guys to watch, and then we'll be all done.
What you are about to see is DNA's most extraordinary secret. How a simple code is turned into flesh and blood. It begins with a bundle of factors assembling at the start of a gene. A gene is simply a length of DNA instructions stretching away to the left. The assembled factors trigger the first phase of the process, reading off the information that will be needed to make the protein. Everything is ready to roll. 3, 2, 1, go. The blue molecule racing along the DNA is reading the gene. It's unzipping the double helix and copying one of the 2 strands. The yellow chain snaking out of the top is a copy of the genetic message, and it's made of a close chemical cousin of DNA called RNA. The building blocks to make the RNA enter through an intake hole. They are matched to the DNA, letter by letter, to copy the A's, C's, T's, and G's of the gene. The only difference is that in the RNA copy, the letter T is replaced with a closely related building block known as U. You are watching this process called transcription in real time. It's happening right now in almost every cell in your body.
Okay, so that takes care of chapter 16, part 1, and we'll see you in class, and then next time we'll go over part 2. All right, talk to you soon.